Peace and blessings. Welcome back to another video. And in this series, if you remember, we have been discussing about the process of replication of DNA. We discussed about the mismatch repair system. In the same context, we are going to discuss today is the certain other repair systems. Okay, so let's start with the first of all, we talk about the direct reversal of the DNA damage. Now we know that DNA damage, it can be caused by a variety of the mutagens. Okay, so that can be the physical mutagen or it can be a chemical mutagen. If we talk about the physical mutagens, we have the UV radiation. So you can see here, first and foremost, this UV radiation, once it falls on the DNA, it can lead to the production of the formation of a pyrimidine dimer. And this formation of the dimer, that will obviously hinder in the process of replication. It will cause the mutations once this mutated DNA is replicating. For that, our system has a direct reversal capability. So that means it utilizes the enzymes like DNA photolase. So now this DNA photolase, how it catalyzes this reaction? This uses light in order to break down this thymine dimer into the individual into the original individual thymines. Okay, so this is a simple example. We call this as the photo reactivation. So the enzyme, it will use the light in order to break the thymine dimer and then thus converting it back into the original conformation. Similar to that, we have certain modifying agents, like we have the methylating agents that will they basically methylate certain residues or certain bases in the DNA. Now there are systems like we have an enzyme methyl transferase. Now this methyl transferase, it will catalyze the removal of this methyl group from the guanine here, and thus restoring back the DNA to its original conformation. Okay, so you can see that here again it is hydrogen bonding with guanine, that is base pairing with the cytosine without any kind of the modification. So these are the examples of the direct reversal. Okay, so simply the enzyme is coming and it is reversing the DNA to its original conformation. Similar to that, we have certain other repair systems, like we have the base excision repair, we have the nucleotide excision repair. Let's try to see the first and foremost base excision repair. By the name itself, that the name says that the base only is removed or it's only repaired. So let's have a look here. If you see here in the DNA, you know that we have first and foremost is the deoxyribose. These are the components or the constituents of the DNA strand. Okay, so in one of the strands we have, here is a deoxyribose sugar. Okay, then it is linked to a nitrogenous base, that may be a purine or a pyrimidine. If it is purine, it may be a adenine or a thymine. If it is pyrimidine, that may be either a thymine, that may be cytosine or the uracil. But in case of the DNA, we have thymine. Okay, and on this side you have the phosphates which are linking the two nucleotides together. Okay, now we can see here in the first case, here you can see is the normal base. Again, here we have the normal base, but here there is an abnormal base which has been incorporated. And now the base excision repair, its function is to repair this damage in the DNA. What it will do is, so first and foremost, it will break the glycosidic bond. So for that we have an enzyme that is called as the DNA glycosylase. Now this DNA glycosylase, as the name suggests, is again, it will break this glycosidic linkage between the deoxyribose and the base. So, and it frees this uracil. Now you can clearly see that it created a apurinic or a pyrimidinic site here. Okay, so that means now this site, it is free of any kind of the base. The next step is the action of the endo or exonucleases, so which will basically cleave here. So it will make the cleave here you can see from the red dots. Okay, so these makes it creates basically a 3' hydroxyl group, so that the polymerase comes and it is going to polymerize in that direction. We know that polymerase, it comes and it will polymerize from 5 to 3 prime. So it has to there is a necessity that a 3' hydroxyl group, it should be there so that it can catalyze the polymerization. So now the polymerase, it will come, and finally the ligase, it will seal the nick. So that is simply that's basically the crux of the base excision repair. So we have, and it has been in the literature, it has been mentioned that we have 11 different DNA glycosylases. So that means there is a specificity when we are talking about the base excision repair. Okay. Now let's move on to understand the nucleotide excision repair. Now earlier it was just the excision of the base, but here we are going to discuss about the nucleotide excision repair. And in this case, we have certain proteins which are involved. So we have basically four major proteins which play a very significant role in the nucleotide excision repair. So we have UVRA, UVRB, UVRC, and UVRD. So first and foremost, this the dimer of, we can see that we have two molecules of UVRA and two molecules of the UVRB. And wherever there is a distortion in the DNA, so first and foremost, this UVRAB hetero, now this is a tetramer, and now the purpose is to locate this distortion. So once it locates the distortion in the DNA, what happens next is the A subunits, they are released. So the main function of A was to locate the distortion in the DNA. Afterwards, the B, it is bound to this distorted region. Now this B, what it does is that it basically docks another protein to the side, that is the UVRC. Okay. So always remember when we are talking about the UVRB, this has a helicase activity. Okay. So that means it kind of, it creates a bubble like structure here. Okay. Then afterwards, this B, it will lead to the recruitment of another protein, the UVRC. And with that, the B is released here. You can see one of the UVRB is released, just one B is there, and with the recruitment of the UVRC. Now this UVRC, it basically excises again. Okay, so it will excise. So you can see that there are two excisions. So one excision is created, that is either 4 or 5 nucleotides, 3' to the distorted DNA region. So you can see that from this side, only 4 or 5 nucleotides, there is an excision made by the UVRC, and on the 5' end there is 8 nucleotides. So starting from this point where there is a distortion, 8 nucleotides ahead, there this UVRC, it will create a excision. So a nick is created. Now afterwards, after the UVRC, it does its job, it excises the DNA. Now afterwards, it leaves the strand. The next, again, the main function that is done by the, so afterwards the UVRD, that has a activity that it will basically remove this fragment, this segment which was cleaved by the UVRC. Then again, we have the DNA polymerase and DNA ligase. So the polymerase, again, as I said before, so we need to have a 3' hydroxyl group so that this will be used by the polymerase, DNA polymerase. It will come and it will go on synthesizing the new strand. And finally, the sealing is done by the DNA ligase. Now here we have to remember that the polymerase, it uses basically the strand which is complementary, which is already there. So there is no mutation, no distortion was present in that strand. So it uses that strand as a as a template strand, and it basically synthesizes a new strand without any kind of the mutation. So that is how the nucleotide excision repair works. Now there is another thing about the nucleotide excision repair. It is that so wherever the polymerase is, so if you remember in my previous video, we have already discussed, when we were discussing about a mismatch repair system, we linked that to the process of replication. So in fact, I often say that we can say mismatch repair system, it is coupled to replication. Okay. Similar to that, you have to remember that when we are talking about the NER, the nucleotide excision repair, we couple this to the transcription. So we say this is the transcription coupled repair system. So wherever there is the RNA polymerase, it stalls, that you will see that it is linked to the process of the nucleotide excision repair. Okay, so that was another point about this. In the next, we will then ahead talk about a next topic.